Before starting this video, I just want to mention, if you find the pace of the video to be slow, you do have a feature called YouTube playback speed. Please use it if you want to. तर बिचारा हल्का मेहनत गर्दै हुनका दाइको लागि. Anyway, हामीले in the previous lecture, I mentioned about simple squamous epithelial tissue. In this lecture, we will be dealing with simple cuboidal epithelial tissue. Okay, so simple cuboidal epithelial tissue is that tissue where the cells are obviously arranged in a single layer because the tissue is simple. But the cells that are arranged in a single layer are cube-like in structure with centrally placed nucleus that is round in structure. And the epithelium is obviously present on a basement membrane. So, these epithelium, that is simple cuboidal epithelial tissue, where is it found? And on the basis of where it's found, what is its function? So, simple cuboidal epithelial tissue forms the germinal epithelium. Now you might be wondering, what is germinal epithelium? So germinal epithelium is that epithelium that gives rise to germ cells. I definitely have not made myself much clearer. So what's germ cells? Germ cells are nothing but gametes. And depending on whether you are a male or a female, the gametes are either a sperm in a male or an egg in a female. Germinal epithelium forms the lining of the wall of testes and ovaries. Germinal epithelium found on the wall of the testes through the process of gametogenesis. Gameto means gamete and genesis means generation. So, through the process of gametogenesis, germinal epithelium present or forming the wall of testes gives rise to germ cell, that is sperm. And through the process of gametogenesis or by the formation of gamete from germinal epithelium of ovary, egg is formed. Okay, you can also find simple cuboidal epithelium in the front, on the front wall of your lens. So the human eye consists of lens, we all know that, which is a biconvex lens. The front wall of this biconvex lens is lined by simple cuboidal epithelium. Tuck, 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 tuck. Yep. Next, number C. Simple cuboidal epithelium is also found in some glands and the ducts of those glands. So on the basis of where the simple cuboidal epithelium is found, we can determine its function as well. For example, here, or the presence of simple cuboidal epithelium in germinal epithelium shows that simple cuboidal epithelium is responsible for gamete formation. In front wall of the lens, the tissue provides protection to your eye lens. And in glands, this tissue plays the role of secretion. And by lining the ducts of those glands, the same tissue provides support. This function right here is important because squamous epithelium is not responsible for secretion. It's the cuboidal epithelium and columnar epithelium that are responsible for the process of secretion in glands. Okay, the same simple cuboidal epithelium is found in human in various modified forms as well. So we'll be dealing with the various modifications of simple cuboidal epithelium. Modification number 1 is your Bros border simple cuboidal epithelial tissue. The name seems long, but not to be intimidated by, because it's really simple. Just break it into words. First is simple cuboidal epithelial. What's simple cuboidal epithelium? This right here is simple cuboidal epithelium. So it's nothing but just that with slight bit of modification. So here are your cube-like cells arranged in a single layer, because it's a simple cuboidal epithelium lying on a basement membrane as every other epithelial tissue. The only modification that is present is that the free surface of the simple cuboidal epithelial tissue, the free surface is the surface opposite to the basement membrane, is Bros bordered. That is, takes the form of finger-like shape, or takes finger-like form. Wiggle, 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 wiggle. This right here is your Bros border. The border forms Bros-like structure. What's the advantage of having Bros border epithelium? This increases the surface area of the free surface. Increases surface area. And what's one significance of having greater surface area? It's none other than absorption. The more the surface area, the more the area for absorption, or more absorption capable area. जति धेरै विग्ली 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 भयो, जति धेरै ब्रस बोर्डर छ, त्यति धेरै सर्फेस एरिया भयो र त्यति धेरै एब्जर्प्सन हुनको लागि एरिया प्रेजेन्ट छ. Okay, so यस्तो epithelium, where is it present? It's present in the proximal convoluted tubule of nephron. So this is all you need to know about simple cuboidal epithelial tissue. You could just end the video right here, because the next thing that I'm gonna tell will come again in the chapter of excretory system, but I recommend you to learn it right now itself. And that is nephron. Nephron. Okay, nephron. Nephron. The first part of nephron is your renal capsule. The renal capsule consists of two parts. So this part is your renal capsule. Renal capsule. The cup-like structure that is present in the renal capsule is the Bowman's capsule. And the capillary ko juppa that is inside the Bowman's capsule is glomerulus. The Bowman's capsule gives rise to convoluted tubule that is known as your proximal convoluted tubule, PCT in short. It's a tubule and it's convoluted, that is highly coiled, and it's present at the proximal end of the nephron. Then you have loop of Henle, descending loop of Henle followed by ascending loop of Henle. Okay, which gives rise to convoluted tubule again. This convoluted tubule is present at the distal end of nephron, so also known as distal convoluted tubule, and the distal convoluted tubule of different nephrons, they end up in a common duct that collects urine from all those nephrons, known as the collecting duct. So this right here is your loop of Henle. This is your distal convoluted tubule, and this is the collecting duct. Why did I draw the diagram of nephron? Because the different parts of nephron consist different type of epithelial tissue that you need to remember. For example, the renal capsule consists of Bowman's capsule. The Bowman's capsule in its parietal wall. Parietal wall. What's the parietal wall? The outer wall of the Bowman's capsule is known as the parietal wall. Consists of or is lined by simple squamous epithelium. The proximal convoluted tubule is lined by this, as I mentioned here, broad border simple cuboidal epithelium. The loop of Henle has two parts. The thin limb, or the tube that you can see right here, that has narrow lumen, and the thick limb, which is the part of the loop of Henle that has wider lumen. The thin limb consists of simple squamous epithelium, and the thick limb consists of simple cuboidal epithelium. Distal convoluted tubule consists of simple cuboidal epithelium, so does collecting duct. Now you might be wondering, oh my god, there are so many parts and so many epithelial tissue to be remembered for a simple nephron. No need to worry. Really simple to understand if you get the concept properly. This is the concept. Here's your nephron. The different parts of the nephron consist of different types of epithelial tissue. But here's an easy trick. The renal capsule consists of Bowman's capsule. Bowman's capsule is where the process of filtration during urine formation takes place. For filtration to take place, stuff has to pass through a surface. The surface needs to be thin, very thin, for diffusion to take place. And the thin epithelial tissue through which diffusion can take place is none other than simple squamous epithelium. So it is the simple squamous epithelium which lines the parietal or the outer wall of Bowman's capsule. Next, in loop of Henle, the thin limb, since it has very narrow lumen, only simple squamous epithelium can adjust in there and form the lining of the wall of the thin limb of loop of Henle. That's why that's the tissue found right there. Only these two places, one where the epithelium needs to be thin for filtration to take place, and another, the thin limb where the lumen is very narrow, so only one type of epithelium can stay there, and that is the simple squamous epithelium. So those are the two places of the nephron where simple squamous epithelium is found. The rest everywhere you have cuboidal epithelium, simple cuboidal epithelium everywhere. Now, be it proximal convoluted tubule, distal convoluted tubule, thick limb of loop of Henle, collecting duct, all of them have simple cuboidal epithelium. But there's a slight modification in proximal convoluted tubule. In proximal convoluted tubule, under simple cuboidal epithelial tissue, broad border epithelium is found. This is also easy to remember because in urine formation, you will come to know that during the process of reabsorption, which is the second process of urine formation, maximum reabsorption takes place through proximal convoluted tubule. And for better absorption, surface area needs to be more, which is found in broad border epithelium. That's why that particular modification is found in proximal convoluted tubule. And that marks the end of simple cuboidal epithelial tissue. Let me revise it quickly. Okay, simple cuboidal epithelial tissue. It's simple because the cells are arranged in a single layer, and it's cuboidal because the cells that are arranged in a single layer are cube-like in structure. Where is it found? The normal simple cuboidal epithelium without any modification is found firstly in forming the wall of your testes and ovaries. in testes, you have seminiferous tubules, and the wall of the seminiferous tubules is lined by this simple cuboidal epithelium. And what does it do there? It sits there in the form of germinal epithelium, giving rise to gametes by the process of gametogenesis. Secondly, it forms the front wall, or it lines the front wall of human lens and provides protection to human lens, direct protection, by forming a shield in front of that lens. Number 3, it's found in glands and forms and plays the major role of secretion, which is not played by squamous epithelium, as well as forms the lining of ducts of those glands and provides support, since ducts need to be in particular needs to go from one place to other, so it requires support, and this support is provided by simple cuboidal epithelial tissue. Then there's one modification of simple simple cuboidal epithelial tissue, that is broad border simple cuboidal epithelial tissue, where the free space of the simple cuboidal epithelial tissue is broad bordered, which increases the surface area, and increase in surface area means more absorption. In nephron, the capsule, the Bowman's capsule, has its parietal wall lined by simple squamous epithelial tissue. The proximal convoluted tubule consists of broad border simple cuboidal epithelial tissue. The thick limb of loop of Henle consists of simple cuboidal tissue. The thin limb of loop of Henle consists of simple squamous epithelial tissue. The distal convoluted tubule consists of simple cuboidal epithelial tissue, and the collecting duct consists of simple cuboidal epithelial tissue. Oh, sure.